Good morning or afternoon to everyone, and welcome to our webinar, Don't Fear the Robots, Lab Automation for High Throughput Discovery Science, brought to you by OpenTrons and Zymo Research. We're very excited to introduce our speaker, Dr. Carlos Goller from North Carolina State University in Raleigh, North Carolina. Carlos is an associate teaching professor at the Department of Biological Sciences, and he teaches in the Biotechnology Program there. He's really passionate about teaching with technology and including lab automation in molecular biology education. And he's made it his mission to integrate it into courses and undergraduate research experiences. He also has an NSF-funded project to create a network of educators to build case studies teaching students about high-throughput approaches to drug screening, microscopy, sequencing, and other high-throughput applications. So we're really excited to hear him talk about his work in this area today. And I'm also here with Gregory Lee from Zymo Research. Gregory is the automation manager at Zymo, and his team have played a very hands-on role helping Carlos and others develop automation workflows. And finally, I'm Fran Slater. I'm a Senior Director of Strategic Marketing at OpenTrons. And I'm really happy to be able to host this webinar and hear more from Carlos.
Thank you so much for the kind invitation. And both of you, OpenTrons and Zymo, have been really helpful in getting us up and running and sharing a little bit about automation and my passion for teaching this in both the undergraduate and graduate lab. So I'll speak a little bit about using the the phone rings. Okay, sorry about that. So I am happy to be here, and I love learning about automation and teaching about it. And today I'll speak about using the OT2 to teach high throughput discovery. So I really try to emphasize in our biotechnology classes that high throughput discovery is something that can and should occur in undergraduate and graduate molecular biotechnology education. And we can do that by exploring the design for experiments that require automation. And this is all based on the experience of teaching this class, BIT for Biotechnology 479, 579, with the OT2 for several years now. So a little bit about this. My catchphrase for several years has been, "Don't fear the robot." And that's because I think lab automation should be part of molecular biology training. And to do that, we need both the instruments as well as the awareness of how automation helps high throughput discovery science. And I define that as using both instrumentation as well as experimental design to propel advances in generation of new knowledge about active compounds, about cell phenotypes, for example, and learning. And we can learn a lot from how we teach automation to students. And I want to share a little bit about that. So in 2015, I was a biotechnology teaching postdoctoral scholar here at NC State. We are fortunate to have a program where we can cultivate the research and education that postdoctoral research scholars bring and provide the opportunity to design new classes. So in my case, I came and was able to work in the biotechnology program to design a class on metagenomics. So going from a swab, in our case, we swabbed kitchen sinks, to learning about what microbes are there using sequencing technologies. And it's a 3-year postdoctoral fellowship. You learn about course design, you learn about classroom management, you teach and design a lab-based course. And I was finishing up that experience and had the opportunity to apply for a teaching faculty position in the same biotechnology program. And I pitched the idea of designing a class called high-throughput discovery. And our tagline and goal was to engage students in lab and computer-based activities that featured automation and high-throughput discovery science. And to do this, Dr. Stephanie Chen, also here in the biotechnology program, and I designed the course after surveying both faculty here at NC State, researchers in the Research Triangle Park down the street, researchers from Duke University, UNC Chapel Hill, collaborators from Southern Research Institute in Alabama and elsewhere, and automation experts. And we asked them the question, if you were to develop a high throughput discovery class, what would you need? And now what would you focus on? And this helped us design a course and write module and course objectives. So we had a set of 6 or 7 course objectives that we wanted all participants. And I should mention that this is a half semester lab-based course for undergraduate and graduate students from several different majors and programs throughout our campus. And we designed several modules, 7 modules, each addressing different questions. So for example, the first module is, what is high throughput discovery? And module 2 is, what makes a good screen, where we talk about reproducibility, Z factors, and calculations. We were able to purchase an EPMotion EP5075 and 2 Opentrons OT2 instruments thanks to internal grants and funding. And this got us up and running, and we were able to pilot experiments in the lab with students and a fantastic graduate student, Jamie Nosbish. And the class took off. I was hired as teaching faculty in 2015 after proposing that course. Dr. Stephanie Chen and I designed the course with help from Jamie. We launched it and launched at the labs in the spring of 2016. We had, at that time, just the epMotion, and we limited the class to 12 students so that we could work in groups of 4, and 4 students would work in front of the instrument, and 8 other students would work on some what we called quant quizzes, so some of the math behind some of the screening procedures. We really refined the labs in 2016 and 2017. 2018, we were tying into some campus-wide challenges because we realized high throughput discovery required compound libraries. So the National Cancer Institute shared a couple of compound libraries, not huge ones, but big enough that we were able to do some high throughput screening with students as part of the class. We partnered with some chemists down the street, Josh Pierce and his lab, to screen some potential antimicrobial compounds. We used my pet microbe, Delphtia acetovorans, as a case study, and we gave students before COVID in 2018, we did a campus-wide challenge where we gave a first-year environmental science students and staff and faculty and friends of the library, we gave them swab kits, and we said, where do you think this microbe that's able to precipitate liquid gold to solid gold nanoparticles, where does Delphtia acetovorans live on our campus? And we made little stickers and we shared them with the campus and got swab kits. and had participants send samples in, and we screened them as part of the class for the presence of this microbe using at that time real-time PCR. In 2018, we applied for internal funding, and uh, I had the opportunity to write a science, technology, engineering, and math internal proposal with my partner, Dr. Claire Gordy, in genetics, and we pitched the idea: don't fear the robot. Use automation, use OT2s in both the biotechnology and the genetics labs. And we were able to purchase 2 instruments, 2 OT2s with all of the modules at that time, and use them in the class. So now we had the opportunity to really engage all students. Groups of 4, 2 groups of 4 were working on the OT2s. One was working on DEP motion, and we rotated the groups during our 5.5-hour labs. So everyone had a chance to really work with the instruments and learn how to program them, how to some of the challenges when it comes to pipetting certain liquids. And we had a fantastic time with that class that we offered every spring and one fall in 2017. And then in 2020, in the spring of 2020, in the middle of our half semester course, after the first in-person lab, the massive shutdown came about with the pandemic, and we were forced to go online. And I found myself strapping a GoPro camera that I have up there to a liquid handler and to the OT2 and trying to make videos. And I stopped and asked myself, is this going to work? What are you doing, Carlos? How are you going to teach a course based on instrumentation online? And in 2020, in the spring of 2020, we switched to online, and we developed a completely online asynchronous high throughput discovery course I want to talk to you about. And this was with lots of help from OpenTrons and Zymo. And today we're back to in-person labs, but we still are using several of those components from the online experience. So what did we do with OT2s? We designed OpenTrons challenge activities. These were group activities in groups of 4 undergraduate and graduate students from different majors and programs, with the goal of familiarizing learners with the software and OpenTrons operation. To do this, these students worked together to successfully complete a procedure using the OT2. The teams began with food coloring and just calibrating the instrument, making OT2 OpenTrons logos, doing patterns with food coloring at 96 and then 384 well plates. And every week, we increased the level of complexity, which included downloading scripts from the OpenTrons site and using scripts with different plate and tube formats, telling students, well, A6 and H12 have candidate compounds. Cherry pick those and do dilutions, do dose response curves. And at that time, we were trying a couple of Zymo 96 well plate DNA extraction and RNA extraction kits as part of this class and tying in to that metagenomics class that I have been teaching since 2013. So students progressively learned confidence or gained confidence with this instrument. and the software and calibrating it. And then by the end of a couple of weeks, they were able to use the protocol designer from OpenTrons to create their own scripts. And thanks to Jonathan Lamb's help from Zymo and graciously sharing how they approach automation of kits, sharing some of the plate layouts and some of the reagents and racks, how they set them up. We shared that information with students and said, here's a biological question. Can you address that biological question with this Zymobiomics kit or this direct soil RNA kit and the OpenTrons? So this is an example of a software challenge. Software challenge 1. You'll notice that it's in an electronic lab notebook we use. Students work in groups, but they each have to individually complete the challenge in terms of the write-up and the reflection. We have some reflective questions. The first challenge really tells them step by step what's happening. Set up the deck of the OpenTrons with this. You are going to do this with food coloring and create this pattern. And the goal here was challenges that encourage students to explore the OT2, familiarize themselves with calibration, with running scripts, with some of the troubleshooting, with adding tips, the different consequences of not having the right volumes or not thinking about the placement of things. And it was stress-free. It was food coloring and lots of reusable plates, and the groups worked on it. And then after an hour, we would rotate to the next station, and groups were encouraged to work together and share. For example, group 1 encountered this issue. We fixed it this way. What will group 2 do? And then by the time we get to software challenge 4 in week 4 of this 8-week course, you'll note that the software challenges become a little less prescriptive. The goal is stated, but then we just say, maybe it was because Carlos was lazy, maybe we just say, visit the protocol page here, download the script, and now edit the script to do this, or to do this number of samples instead. And the goal here was to increase the level of difficulty, promote independence, and allow students to explore some of the higher complexity tasks that automation can enable us to do. So by increasing independence and providing students practice and allowing that time for them to explore and exchange ideas with peers, troubleshooting became part of the process. We laughed when liquids and tips fell. We shared strategies with peers, and the deliverable was always as part of those electronic lab notebook entries. We always had a deliverable that was an image of hopefully success, maybe a plate with food coloring, or maybe a plate with bacterial cultures. But even if it wasn't successful, we encouraged students to take pictures of workarounds. And there were some really fantastic workarounds. And for example, in this screenshot on the right, we have an OpenTrons protocol, and we encourage students to complete this script with minor modifications and share their successes. So when it came time to combining the OT2 with Zymo research kits, we used the Zymo Biomics and MagB96 kits to engage students in the design of unique experiments. And this included scripts and clever solutions to address novel biological questions. So again, in groups, with the help of a fantastic postdoctoral scholar whose expertise is in plant genomics, Dr. Carly Sjogren and I designed the online version of this course. So again, interdisciplinary groups. We were lucky to have 2 or 3 instructors, so a postdoctoral scholar, myself, and a graduate TA, and we each took on the task of being a coach for a group of 4 or 5 students. And what we decided was to do a Jove Journal of Visualized Experimentation style manuscript based on the design of an experiment using the OT2 and one of the kits. So we told students, and throughout the online course, which was completely asynchronous with some in sync or synchronous Zoom open hours for students to talk to each other and talk to us. Throughout the online course, we offered students articles that used automation, and these are articles from the journal Jove. And these include, if you haven't seen them, they include videos with the researchers walking through a procedure. And it allowed us to bring instrumentation we don't necessarily have access to, so new technologies, new liquid handlers, as well as bring liquid handling and robots and really great science online. So students watched the Jove videos throughout the semester. We had one every other week. And then their task was to address a biological question that the instructors came up with using one of the kits, Zymobiomics or MagB96 for DNA or RNA. We told them about the protocol designer, and we helped coach groups of students to design and develop and write a manuscript that they had to submit at the end of the semester. So this allowed us to really have fun with the process of automation and leverage the protocol designer as well as Zymo kits and expertise from Zymo, from the Zymo automation team, to have students design really novel applications to, for example, screen bacterial isolates after extracting DNA and setting up digital PCR or real-time PCR, extracting RNA in 2 different conditions for bacteria treated with heavy metals. People got really creative and produced detailed scripts as well as articles with the steps and the reagents. And this was all thanks to the availability of the OpenTrans protocol designer and these kits from Zymo. That I'm going to transition over to how Zymo allowed us to do this.
Thank you, Carlos. Actually, basically, I thought I'm excited. I wonder if I can participate in your class and maybe speak to your students. I always speak to you, and you're lovely, but I'm excited to also be able to speak to your students. But maybe I can take a moment and I can speak to you all about, well, we mentioned Zymo research. So what is Zymo research? Zymo research, just as a sort of general introduction, we're a biotech company. We were known for specializing in DNA, RNA extraction, but we've also gone into the fields of microbiomics, epigenetics. As Carlos had mentioned, we're very much in this field of microbiomics and metagenomics. So our real goal is to be able to provide tools for researchers in numerous number of fields, right? Whether it be basic research and discovery or application research, clinical diagnostics, even down to personalized medicine applications. fundamentally, we're an innovation company, and we simply use biochemistry as our tool to find applications. And so we've expanded that to become not only knowing sort of the research and the ability to apply these tools, but we also realized, well, if we're going to sort of get these tools out to researchers, we need to get them out in the best way. So we developed even as a kind of true manufacturing company, which is not only where we're designing and manufacturing the biochemicals utilized for our assays, but even developing into disposables and full kits. And kits not just for extraction, but even down through the real-time PCR or library generation. Well, as part of that expansion and part of that need, we realized we need to help people get into automation. Not specific to automation, but really a methodology for high throughput processing. So we just usually just refer that to automation. In a way, what Carlos was speaking about, we saw the very definite need for our field, the biotech field, to expand in this direction. This is fully independent, so maybe like any good scientist, we see that there are two independent experiments that led to the same conclusion. But the, I guess, benefit to Carlos is that he gets to teach his class. And for us, from our end, we have to work with customers where they're at. And I would never say there's a lack of understanding, but there's oftentimes a lack of familiarity. So for us, our aim as part of the Zymer Research Automation team is really to create simple, user-friendly, something that's readily accessible for customers to easily carry out their experimentation on an instrument. In this case, we're for Carlos and his class, that they've essentially started working with OpenTrons OT2s. So where do we start? How does our process work? Well, one, we have the benefit of having kind of very robust and flexible biochemistry or chemistry. We know our kits very well. And in fact, my entire team, although, yeah, we're composed of engineers and scientists of multiple backgrounds, we specialize and devote a lot of time understanding our particular chemistry and kits. And that helps, right? Because then we know what kind of shortcuts we can take or how we can modify our protocols to get the most robust application on a high throughput system. Very basically, maybe to step back, is whenever we look at a project, we always define it in three major steps. One is the scripting, which is the actual programming. So I think Carlos touched on that very well. Getting his students used to the program, getting used to the OT2's protocol designer, and that's essentially what we also do. We need to spend a lot of time planning and scripting and making sure that what we desire for the OpenTrons OT2 to do, we've planned that very well through our scripting process. The next step on that is implementation. So that's, again, what Carlos is doing with his class. He's getting them working with the actual instrument. When you tell an instrument to pipette a certain amount of liquid, does it do it? And does it do it from the right trough into the right well? So that's just part of the process. And for us, we go through one more step. It can kind of be said that Carlos also does that with his group in terms of experiments, but because we are defining an assay kit, we go through a process called verification or validation. We utilize the term verification because verification means that
we have particular metrics that we want to hit from our kit on the OT2 system, but it doesn't necessarily validate for a particular experiment. So that's really the only difference between the two terminologies. So what does this all mean? After we've done scripting, after we've done implementation, after we've done verification, how does this help a customer? Well, even though we could basically hand over a script and hand over an assay kit, we realized that is still a very intimidating process. So we went back and we realized there's a need, a want, and a nice to have from customers. Yes, the customer needs a script. Yes, the customer needs a kit. And they want to make sure that that script and kit work on a particular machine. But we realize it's also very nice to have a sort of designed out protocol and a designed out presentation to give to customers. I think that's what Carlos was alluding to, is that when we provide a lot of our support, we try to not only help customers and students understand what they're about to do, but have a sense of familiarity, have a sense of, I can do this, I can achieve this. We like to have things presented in a way that makes everyone feel that science is accessible, and that the high throughput field is accessible, which is why we're more than happy to be working with Carlos's group, as well as the OpenTrons team, because we have the same goal of accessibility, making science accessible to everyone. And so what does accessibility look like? We always define it as capability, capacity, and consistency. This is no different than whether you're working with high throughput applications, or really when you're just doing science on the bench. But we want to make sure that we grant customers capability on their instruments by providing all the resources. Making sure they have the capacity, meaning that if they have a large number of samples to process, what does that look like? Maybe even beyond just having one OT2, maybe they need to have a few OT2s set up. And consistency, meaning that whether day in, day out, you're still getting the same results, you're still getting the same robust values and things that you've seen day to day and between user to user. So, and a lot of this isn't just dependent upon the chemistries that we produce. It's not just dependent upon the protocols we produce. It's very dependent upon the hardware of the machines. And so maybe I'll turn it over to Fran, and she can go over this robust system of the OpenTrons OT2.
Great, thanks, Greg, and thanks also, Carlos, for the great presentation. It was fantastic to hear not only your enthusiasm, but obviously the enthusiasm of your students really came through. So yeah, we'll just start off explaining a little bit about OpenTrons for people who maybe haven't heard about us. So we make robots for biologists, to put it very simply. Not just robots, but the robotics, the software, and the community tools for life scientists. And as Carlos has already mentioned, our flagship robot is the OT2, which is an accessible benchtop liquid handler that's really built for life scientists, especially molecular biology. And this is what the OT2 looks like. It fits on half a lab bench. It's compared to normal lab automation and liquid handling technologies, it's incredibly affordable. So Gregory just mentioned accessibility, and he mentioned that that's what we're all about, and that's absolutely true, right? One of the things that prevent people from getting into lab automation is the high upfront cost, and then also, as Carlos mentioned, the fear, right? So that's something that I think we're all very committed to, is really opening up the possibility of lab automation to a broader audience. So the OT2 has high performance comparable to robots that are much more expensive than it. It's also flexible. I'll show you a little bit about I'll show you on another slide a little bit about the modularity of the platform, and it's open. So our software is open source, and it's consumable and reagent agnostic. So you can use any reagents and any labware on there. It's also powerful. So Carlos mentioned the protocol designer, which I'll show you in a second, but we also have a Python API. So for complete control, you can really script the robot to do anything you want. So you can either use the protocol designer, which is a GUI, or there is advanced scripting capabilities. And it's supported, right? So, we have a team of field application scientists and support folks and applications engineers who are there to really help people get up and running. And then, of course, we also partner with Zymo and Gregory and teams like that to help customers get up and running with particular reagents on particular robots. So I'll just show you a little bit. This is, what do people use the OT2 for? Well, a lot, and there's a really wide range of applications that you can use the OT2 for. But the applications that Carlos has been talking about, so really molecular biology and microbiomics and NGS, but those are the really popular applications. So there's a list of applications that are most frequently used for the OT2, and really genomics and related applications are our kind of sweet spot. I mentioned the modularity of the system, and this is just a cute graphic that shows this in real time. So there are what we call deck positions on the robot. So there are 11 places that you can put things on the robot, and those can be hardware modules like a thermocycler. There can be tips and labware like a 96-well plate. Anything that has a standard SBS footprint can fit on the deck, and you can move those things around depending on your application and your workflow. In the top right-hand corner, the only thing that isn't moving is the waste, and so that's where the tips get ejected. As I mentioned, those deck positions are for SBS format, consumables and labware and so on. And all of this, everything can be removed as well. So all of that, all of the consumables, but also the hardware, such as thermocyclers or magnetic modules, they can all be removed and moved around. And then pipettes, we have a variety of pipettes that could be put onto the gantry. It can hold up to 2 pipettes at the same time, and you can choose the pipettes that you want based on your application. So we have single channel options and 8-channel options in a variety of volumes, and they're sort of all interchangeable with each other. And so we also have a variety of hardware modules. I'm just showing the magnetic module here because it's one that is really commonly used for magnetic bead-based applications such as Zymo's microbiomics kit and others. So it's difficult to see here, but it has an array of magnets that kind of pop up in between the wells on a 96-well plate and engage and attract the magnetic beads, and then when it's appropriate time, the magnets kind of disengage and so on. It's a cool design for doing magnetic bead-based extractions and similar work. And finally, I just wanted to show a little bit about Protocol Designer. So this is the GUI that Carlos's group use in their experiments. And it's a code-free GUI software that helps you to build protocols. I will show a quick demonstration video of it in a sec, so you can. But generally, it's pretty intuitive. It's all visual and so on, and you can lay the deck out how you want, select hardware and liquids, and then you can program the pipettes to transfer, the right liquid from the right well, right trough to the right well at the right time. And so here's a quick 30-second video to show what that looks like. Okay, that's about it. I just wanted to say thank you to Carlos especially, and also Gregory for joining us for this webinar and for giving such an interesting presentation. And also I'd like to thank all of the students that have gone through Carlos's course and have also obviously contributed to this work in such an important way. So thank you so much, everybody. And now we'll open it up for questions. Okay, so welcome to the Q&A session, everyone. We have 5 or 6 questions already that people have entered into the Q&A box, but if anyone has other questions, we have plenty of time to address them. So just type them into the bottom left-hand corner and we'll get to those questions as well. So I'll pick a random one to start on. So Carlos, start with you. What has been your students' feedback regarding the courses that you've been talking about?
I think this is a tough question. It depends. If we have undergraduate and graduate students, some graduate students are now go back to their labs and ask, for example, I need do we have access to someone on the floor with automation? Can we get an open trans? Can we look into automating this process because we do it routinely? As some undergrads go out and get jobs in biotech and come back and say, guess what? I've been using this or that instrument and I've been doing this. So that's really great. We've also have the perspective of some students loving the challenge of the scripting and thinking about the steps. So we've had a couple of people mention, oh, I love the challenge and now I'm learning more about Python. They like the graphical user interface, but they found that it was more powerful to be able to define things exactly how they wanted.
Yeah, very cool. And Python is becoming like a really, really like a really dominant coding language. So I think everyone should be learning Python at some point in their undergraduate education now. Okay, let's jump to the next question, and maybe I'll. It's more of a technical question about the Zymo kit, so I'll put this one to Greg. What's the maximum throughput for DNA or RNA extraction using Zymo kits on the OT2?
Yeah, that's a great question because it talks about basic throughput. On the OT2, we basically design it so all of our kits will be
walk away at least 24 samples. Some of them can do 48 and walk away, but primarily we just say generally, right? It's going to be 24 samples. But that can always change depending on what everyone wants to do or manipulate in terms of the chemistry. But I would also say that OT2 is certainly capable of doing 96 samples at a time. Just the sort of limitation is, you kind of run out of tips. So you need to add more tips or this or that. So the capability of the machine and the instrument is there, for typically where people want to process about 96 samples. But if you kind of want to just press a button and just let the machine do its thing, it's typically minimum 24.
Excellent. Thanks, Greg. One again for you, Carlos. So you have an NSF-funded grant to sort of build a network of educators that sort of interested in some of the aspects that you were talking about in terms of building courses and curriculum. How can people join that network to collaborate on that?
Oh, that's really good, because we have ongoing efforts to try to expand the network, and it's going to be HITS version 2, and I'll put the website is go.ncsu or North Carolina State University.edu/HITS, HITS. So Sabrina Robertson came up with a really awesome acronym, and that takes you to a QUBES page. That's QUBES. So Quantitative Undergraduate Biology Education System. And that's where we have a community of people developing resources, case studies to bring high throughput into a variety of classes. And for me, it's fun to play with. High throughput is automation or sequencing, but there are several other ways of doing high throughput science.
Excellent. Thanks so much, Carlos. This is more of a general question. I think it would be good if everyone sort of answered from their perspective, sort of in turn. But what's my first step if my lab wants to implement automation? So, Carlos, I'll start with you as someone who has implemented automation.
Oh, and this is gonna sound silly, but we do it in class. I'm a big fan of flow charts and diagrams, and just go find a whiteboard or find paper, draw out the main steps and of the procedure, and think about what can this be automated and in a way that will save you time and can be used over and over. And then if you have access to a liquid handler, now that you have your main steps and you're thinking in terms of where can I put this volume of liquid that I have to distribute into this number of vessels, then bring out the food coloring. That's when you use food coloring and ethanol, when you have ethanol stuff or a reagent with ethanol or glycerol, and test it first with food coloring. And for us in a teaching setting, that's really great. We often take a protocol, download it from Zymo, or download a protocol from QIAGEN and say, okay, we want to automate this. These are the main steps after you drew them out. Let's load up the liquid handler and do it with food coloring.
Yeah, food coloring is that's such a top tip. I think glad you mentioned that. Greg, what's your perspective on that, implementing automation?
Yeah, I guess from like automation kind of specialist or technical help, like Carlos said, drawing up the flow chart's actually really important, because I think oftentimes when we when we go into this idea of like automation, we immediately think, okay, what's the instrument I need? and which is fair. It's a fair thought, right? Because that's the biggest investment typically. But I would say, chart out the whole process that you're trying to achieve, and figure out where your bottlenecks are, right? Because if you're really, there's one thing to say like, I want to do automation, but I guess just from our backgrounds, like, well, wanting is great. And that's where you can get into experimentation and just basically say like, okay, well, at least here's my flow chart and here's what I can do with the liquid handler. But from the aspect of, well, what is the need that the automation is going to meet? Well, that's where these flow charts and understanding where your bottlenecks are going to help you. And can the liquid handler do that for you? It's great that we can, place a Zymo kit onto an Opentron's OT2, but if that really isn't your bottleneck, right? If you're only doing, let's say, like 2 samples a week, maybe putting it onto, an Opentron's robot, to meet a need isn't necessarily there. I always recommend, from customers or those that come and ask us is, what is the need that the robot needs to meet for your lab or for your business? And, then how to ask the right questions, right? Ask the right questions from the instrument vendor, from Opentrons, from Zymo. And I would also say it also helps address not just sort of what kit I need to place onto the instrument, but it opens up what the capability of the instrument can do, like even from sample handling, like distribution of the sample into a plate. Certainly, doing extractions using a Zymo kit, but even your library prep, cherry picking, all those sort of things can be opened up by using the instrument. So I'm going to turn it over to you, Fran. Like, what's your thing?
I think definitely like to analyze those bottlenecks and to start small. Like almost all automation is somehow modular, right? So you don't have to go for a 6-figure setup, to get started. Wouldn't that be great if everyone had that budget to play with? But you can do it step by step. I also think that looking for lots of help is really important. So Greg mentioned, that Opentrons and all automation vendors should be able to give you a significant amount of help in terms of analyzing what it is that you want to automate it and giving you advice about how to do that, and whether you should do it, or what the pros and cons are, timing studies, all these things to really give you some information about what that automated solution would look like. And I think also like from the community, so Opentrons is we're very big into into community sort of an engagement and involvement, and that's true for people that want to start automating as well. There are lots of forums out there and lots of people who are already automating what you want to automate, and so then I think that's really important to connect with those people and get their perspective on how to do it, when to do it, and so on. Okay, excellent. Okay, so we have a couple of OT2 specific questions, which I could probably answer. So the first one is, what level of programming do I need to know to operate an OT2? So a little or a lot, I think, is a sort of answer there. And I will also get Carlos and Greg to weigh in, knowing that they obviously have worked extensively with an OT2. But fundamentally, we offer 4 different ways to kind of get your protocol done. So the most introductory way is to use our protocol designer GUI, which Carlos mentioned using, and which I showed a little bit of a short demo in the talk. It's GUI based. It's very sort of friendly to use, and it's a good way of kind of beginning to kind of play around with the robot. And on the opposite side of that, we do have Python API. So you can, if you are familiar with Python, and if you really want to get, a very sophisticated level of control over the OT2, you can write your protocols in Python. And that enables you to do almost limitless things with the OT2, right? You can really create very bespoke protocols that way. We also offer 2 other things as well. So we have a protocol library where you can find protocols that other people have written, either OpenTrons protocols that our science team has written, or protocols submitted from the community. So they might not exactly kind of do what you want. That it's, obviously a lot of people have specific needs, but they're a good base. They're a good starting point. People find them good to review as a starting point for a particular application. And then finally, we have an FAS team, field application scientists, who will write protocols for you. That's a paid service. So, obviously it depends on how you want to do it and what your budget is, but we provide kind of consultation and scripting for you. But I'll ask maybe Carlos and Greg to weigh in on programming the OT2. Maybe Carlos, do you want to go first?
I think in the classroom setting, it was really good to have the web-based. I say we first started with the download, the one where you can find a protocol on OpenTrons, change a couple of set parameters, and download a script, and then feed that script into the OT2. I think that was a really good way to get some confidence that you can run something on this instrument, and it's visiting a website and sharing some stuff, and sharing a script with the robot. And then the protocol designer gets more into the design of the experiment without having that programming. And then when people start running into limitations of, I want to do this or I want to do that, that's when you always have the adventurous person who wants to open that file with the text editor and start messing with it. And I think some people really love that challenge, and some others just want to get the protocol up and running or modify, and they can do it in really creative ways with a GUI, a graphical user interface.
Yeah, I mean, I probably have not much to add to what you all just said. I think it really ultimately depends on the user, right? Even if you have no programming background at all, you can operate OT2 because there's so much resource available to you. Like you said, but if you don't have any programming skill, I do highly recommend, either reaching out to OpenTrons or to like a reagent vendor, like for instance, Zymo, we actually have the OpenTron scripts that we can provide, but also like you said, there's the protocol database. That's a great place to start because at least at that point you can find something that's familiar to you, and you can kind of just see sort of the map. And as you get more familiar with Python, you can change things and alter things to meet your particular need. But to sort of hit the kind of extreme side that Carlos was saying, like people who like to just mess with the OpenTrons, like I know that there are people who like 3D print sort of like modules for the OpenTrons, and then they use a lot of Python coding to just to make this system sort of custom to their particular need and tailor it to their particular needs. So I think you just get the extremes just because, and I think it very much demonstrates the versatility of the system.
Yeah. Excellent. Thanks both. I think just because we have 1 more minute left, I think we'll just do one final question. So is there a reason why cell transfers, mammalian cell culture cells, are not emphasized in OT2 applications? And I can take that, but I'll also ask. and there is one more comment that I will make sure that we get to after this as well. But I can answer that question. So, I mean, we do have some protocols around cell culture and transfers and so on. It's not one of our more popular applications. Obviously, we do have a HEPA filter, but it's not, the OT2 is not naturally a contamination-free environment. You can add the HEPA filter, and so that helps. But we do have some protocols around that. So if the person who asked that wants to contact us, info@opentrans.com, we can see what we have that could support you. And just we're out of time, but I wanted to read this that just came in. Just a comment for Dr. Goller. First of all, go Wolfpack. Yes, Biochemistry 2010. As an automation scientist, it's great to see automation being included in science education. Lab automation is a skill I picked up on the job, but it's becoming an increasingly critical enabler of biotech. Kudos for giving your students the advantage of exposure to this aspect of science. So I think that's a great way to end. But thanks so much, Grace, for that comment. Carlos, any final words?
Oh, yeah, I think it goes back to that don't fear the robot. If we can get people realizing that the instrument may not do what you want at first, but you can go back and forth with it and learn really cool tricks. And then once you have a script that runs something, it's really fun.
Yeah. I think that's a perfect. I think we did have one more question, which we didn't get time to answer. But if we can pick that up afterwards and answer via email. But that's a great note to end on. So thank you so much, Carlos. Thanks so much, Greg, and thank you so much everyone who joined and took part.
Thank you all.